public health emergency. As a point of information, we will not be hosting a town hall on Wednesday, July 7th. We will resume on July 14th via Zoom with updated login and dial-in information to follow. Today, Dr. Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, and Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to development and the validation of tests for SARS-CoV-2. Please remember that during this town hall, we are not able to respond to questions about specific submissions that might be under review. Now, I give you Toby.
Thanks, Irene. Thanks, everyone, as usual, for joining us this week. I have a couple of updates, and then I will go through some of the questions that we received by email before we start the live questions. So first, I know we've had a lot of questions about the decision summary for the BioFire de novo. That decision summary has now been posted on our website. It was posted last week, I believe, on the 25th, and that can be found on the de novo summary page, as well as the, with the BioFire de novo authorization itself. And then I also wanted to update that we just recently added three tests, 3 EUAs to the pooling and serial testing amendment. So that is the amendment that we put out recently to allow for a streamlined addition of pooled serial screening to previously authorized RT-PCR tests. So an email went out earlier this week about the 3 tests that were added, and those can also be found on the EUA page, the molecular EUA page, where they'll be noted under the amendment itself, as well as the authorization documents for those 3 tests specifically that will be available, or that are available rather, on that page. So to get into the questions that we received by email, I do want to note, as we have before, that we've received some questions that are a little too detailed or test specific or case specific to address on the call. So we won't address those individually on the call. For those questions, we will try to send a response in writing within a few days. And if you've submitted a question and don't hear it addressed on the call and don't receive a written response in the next few days, please feel free to reach back out to the CDRH-EUA-templates at fda.hhs.gov mailbox for an update. So the first question that we have today is regarding an EUA submission for a professional use diagnostic assay where they're pursuing a clinical performance study using retrospective samples stored in viral transport media, and as well as collecting prospective samples in VTM to complement that data set. However, they also note that they have a proprietary assay buffer that they intend to supply with the kit, and they're asking about matrix equivalency study to demonstrate compatibility with the proprietary buffer, and whether they need to repeat all the validation studies or just do a limit of detection analysis as an acceptable matrix equivalency. So generally, the matrix equivalency study should be performed to compare the assay performance in the VTM and proprietary assay buffer. So we would recommend testing be performed in parallel at the same target levels in both simulated and naturally occurring matrices. And to demonstrate equivalency, you should observe the expected proportion of positive results at each target level in both sample types. The LOD can serve as an acceptable matrix study, and if determined to be equivalent, repeating the other validation studies should not be needed in most cases.
I would just add that this is kind of a little bit of a special case, but where banked samples are used for central lab test validation, which has always been acceptable for molecular tests. There are specific potential issues with antigen tests, and we want to work through that. So, but the same thing goes for point of care. So we are accepting bank samples for our point of care and for molecular. Typically, we would like you to use whatever buffer you're going to use in your actual test if there is going to be a transport buffer. This kind of bridging study, cross-validation, is perhaps a special case. We do know that using bank samples, especially bank positives, is going to be important for many now that test positivity rate in the U.S. is going down. And still, say an antigen point of care test, we would like to see, and we've done this before. In fact, the first antigen authorization was allowed completely banked samples, BTM, frozen. But we also recommended that they do at least 5 positive fresh samples, demonstrating good results on those 5 prospective fresh samples, and the intervening negatives that happen with this collection of those fresh positives. So fresh negatives and fresh positives, we expect that fresh negatives are going to be easy to obtain, and complete data set on using bank samples, particularly for antigen tests, is good to establish performance. We just want to see those 5, a minimum of 5 fresh positives, and then the remainder of the fresh positives will be placed into a post-authorization requirement study. Thanks, Toby.
Great, thanks, Tim. All right, the next set of questions that we have is related to antigen studies. Regarding previous statements that we've made on the town hall about allowing inclusion of data from outside of the U.S. for point of care, as long as the sponsor can define how the forum sites qualify as point of care, and asking whether any countries are excluded from that, whether variants which exist in certain countries is a concern for labeling limitations, and specifically asking whether, for example, they could use 80% of data from a point of care site in India, 10% in the UK, and the rest in the US. Then they're also asking about using previously frozen samples for point of care studies, as long as a freeze-thaw study is performed. So it is correct, we have previously stated that it may be acceptable to use data from outside of the US. However, we would want to see that the sponsor has demonstrated that the point of care operator in the OUS setting is adequately representative of a point of care operator in the US. And this could be done by providing a detailed protocol describing how the proposed setting and intended users represent the point of care sites and users in the US. We would want to see that the instructions for use are in English and provided in English to the point of care operator to perform the testing. So that may present a challenge in countries where English is not the primary language. But we would recommend that the sponsor submit a pre-EUA that includes a detailed protocol, including details on the sites, users, educational backgrounds, professions, et cetera, to demonstrate comparability or representability of a U.S. Point of care site. And then the information that is provided in the labeling ultimately would depend on the data obtained in the clinical and analytical studies. We do have information in the viral mutation guidance about expecting test developers to consider the potential impact of genetic mutations and variants that are in circulation in the U.S., as well as a plan to routinely monitor for new genetic mutations and viral variants, and to assess the impact of mutations or variants on their test's performance, considering the potential of a given mutation or variant to impact their test. And regarding previously frozen samples, we have previously discussed incorporating archived specimens into your validation. We would want to see that the testing is performed on your device in the point of care settings with the intended users, and that the supplemental banked specimens should be integrated into the daily workflow of each user rather than included as batch testing. We would want to see that the archived specimens were prospectively collected and have a documented history of previous test results, including all test dates. And we would also want to see that there is well-documented information on the storage conditions of the samples from the time of collection. And then also, as previously discussed, a fresh versus frozen study should be performed to demonstrate that the test has similar performance with both fresh and frozen specimens. And then we would generally also expect a post-authorization study using freshly, prospectively collected samples if frozen samples are used for the pre-authorization study.
And as I mentioned before, thanks, Toby, if it's antigen, we do want to see those fresh collected samples, 5 minimum ahead of time. The importance of how you sample a bank is important. We want to eliminate bias, say selecting only high positives or other such things. And we want to see a normal distribution using bank samples as well. And then back for the topic on rest of world studies for point of care. We recognize this isn't necessarily a preferred method because we really like to see how it works in the U.S. for U.S. authorizations, but we know because of the low positivity rate that it's very challenging to collect samples quickly enough, positive samples quickly enough in the U.S. In regards to the English IFU, I also want to clarify that if users are not fluent in reading and understanding English, that's not an acceptable participant in the study. We've seen some sponsors for some point of care and even home use instruments and tests that have provided local language translations of the IFU that are used primarily by the users, and that is not something that we recommend. We are testing the English language IFU as part of the test, and so it does need to be with folks who understand English. All right, thanks, Toby.
Thanks for adding that detail, Tim. The last question that we have to address right now is also regarding the use of banked samples for point of care validation of an antigen test. This is specifically asking about the use of a set of banked samples that includes all required information identified in the EUA template except for the number of days post-onset of patient symptoms, and asking whether they can still use that cohort of banked samples for initial EUA authorization without that information. So generally, while we have indicated that banked samples may be acceptable, we would need to see that all of the data associated with the samples is available, and that does include the number of days post-onset of patient symptoms. So we would we don't think that this data set is likely to be acceptable since it does not have that information.
Those bank samples could be used for analytical studies, though, perhaps, if appropriate. This is an antigen test question. And one of the challenges of using bank samples that don't have data after onset of symptoms is we do not want developers picking samples based on the CT value. The selection should be unbiased. If it's unbiased and you don't know the days after the onset of the infection, you can end up with a lot of very low positives, high CT samples, and it will not put your device in good light. If particularly antigen tests, we don't have an expectation that 80% low positives are going to be able to, or say 90% or 70% of low positives being able to be detected at a sufficient sensitivity. That we just don't expect that of the antigen technology. It may be easier for molecular to do that, but it also puts your molecular device, particularly if it's a point-of-care device, it might be a little bit less sensitive, at risk if you use such bank samples. So we've clearly seen some examples of developers and test developers that have gone to bank samples and ended up with a skewed sampling because they don't know the days after testing, and then they have a lot of too many, really, for the device to look well in comparison to the comparator test because there was an unexpected imbalance in the number of low positives that were included. So that's the danger and risk of using bank samples. And so it's important for us to know the days for which an antigen test is viable as a test, and so we need that information. It also behooves developers to protect their investment by carefully using banks that record this information. Thank you.
Great, thanks, Tim. With that, I think we can move on to the live questions.
Thank you. At this time, if you would like to ask a question on the phone, please press *1 on your touch-tone phone. Please ensure that your line is unmuted, and please record your name when prompted to be introduced. Once again, please press *1 at this time. And we have a question from Greg Sloblakian. Your line is open.
Thank you. If I could get someone to comment on a NIH-funded study that's out today, it was announced. It basically builds the case for frequent COVID-19 antigen testing. The study basically says that antigen tests, if they're given every 3 days, are on par with PCR tests. And I'm just noting here also that I think there was 43 people in the study who were infected with the virus. So I don't know, Tim, is that something you could comment on? Just the whole, building of this case, that antigen tests are on par with PCR tests.
Yeah, no, exactly. I think this is the full journal publication of this data. It was out as a preprint months ago, and the data was shared confidentially with the FDA before the preprint came out as well. We used that data, which was not done in always in a traditional FDA study sort of manner, but we used that data to primarily inform our recent policy, which allowed serial testing to support the provision of serial testing to support an asymptomatic screening claim for authorization without having any asymptomatic data. So that policy was based on that data, which was available to us months ago. it's the basis for our serial testing program for any assay that showed at least an 80% sensitivity PPA in symptomatic individuals, and had a lower bound of the 95% confidence level of at least 70%. And so we had good indication that the test was performing well enough in symptomatics, and the mitigation for expanding the claim to asymptomatics without data on asymptomatic individuals prior to authorization would be an acceptable risk for the agency. So we allowed that. And then we did have a post-authorization commitment by the companies that use this, that they would do a serial testing program after authorization to demonstrate adequate performance particularly when antigen tests are used in serial testing, 2 or 3 tests a week. There are several antigen tests that utilize this, and we allowed that and then allowed us to make those tests rather than prescription home use for those suspected of COVID, to authorize them based on that paradigm for an over-the-counter, which also would test asymptomatic individuals as well. That particular study was done with only one of the lateral flow tests, and one of them that might be more sensitive because it's an instrumented lateral flow test. And so we don't know that we can generalize that to all developers as far as basing regulatory decisions only on that study. So that's our current thinking. Hope that's helpful.
Gotcha. Just a quick follow-up, Tim. Is it your perspective that PCR is still considered the gold standard for diagnosing COVID-19 infection? And is the FDA still recommending that if there's a positive result from an antigen test, that it be verified by a PCR test?
So there is communications that are out on the FDA website and on the CDC website regarding all those. We support the use of any test that can perform adequately to receive regulatory authorization. There are use cases for molecular, and there are separate use cases, and some of it's non-overlapping for rapid point-of-care tests, many of them being antigen tests. So remote areas that where turnaround time for a molecular central lab test is not ideal. Use of antigen tests or other point-of-care molecular tests may be used, but antigen tests can be produced at manyfold access to what a molecular point-of-care test can be, and often those molecular point-of-care tests require an instrument, whereas a couple of the rapid antigen tests don't require an instrument at all. So that has clear advantages depending on the use case. And so we don't have a recommendation over. Now, of course, we want to know as close to truth when we're evaluating tests as we can. And we believe that central lab high sensitivity molecular assays with an extraction step represent the closest information to truth about whether a patient is infected with SARS or not. And so we typically use that as the comparator test for all other tests in some sample types. And that just allows us to understand, what the real situation is. Let's see, what else that I wanted to add into this discussion. So we clearly see, a value for antigen tests. As far as a follow-up to antigen test results, this is frequently the case with any test. We have specific language in the point-of-care antigen test as well as antigen tests that may be less sensitive than the central lab molecular test about antigen tests actually are presumed negative. And if they are presumed negative, we want clinicians to evaluate whether that negative seems real to them or not. And that's all in the authorized labeling that we have for those tests. You can go to the FDA website and look at that labeling. But with any test, including central lab molecular tests, there can be false negatives and false positives. A lot of the variability is also contingent on sample collection. So, or there could be a mutation that might be affecting an individual patient. We're not seeing any mutations or variants yet confirmed that are significant risks to test performance, but we have seen individual cases in low abundance mutations or variants where a certain test it does fail, and so it does require clinicians, laboratorians, everybody involved with SARS testing to keep that in mind, that they could have a false positive and false negative. We also have said that if you have a very low incident population, no matter what the test is, molecular or lateral flow, molecular has a little bit better specificity, NPA than lateral flow typically. But in either case, you're going to start to see when the incidence of true positives is low, that you may see significant numbers of false positives. And we've also seen and had reports that clinicians move positive patients on an initial result in a low incidence environment into a COVID ward, and that's obviously risky. So we do ask that everybody involved with testing be aware of that. And if the incidence is low and you have a positive, unless that person is already at increased risks, as they're known to be exposed to a confirmed positive patient, that you consider that it could be a false positive and do a confirmatory test, which we would recommend be a molecular test. So hopefully all that is useful.
Yes, thank you.
Thank you. Our next question is from Dr. Gurgis. Your line is open, sir.
Yes, this is about the OTC and the usability studies, human comprehension. We submitted the application. It seems to be applicable to point of care, but then up to the level of the over-the-counter. Since we are approaching testing populations from 2 years old to 99 years old, what does the FDA expect us to do for the usability studies? Can you comment on this, please?
Yeah, so I think what you're saying is for the younger age groups, what do we expect to see for the usability studies? So it depends on whether you're going to have younger patients self-collect, or if you're going to have adults collect the sample. So if you're going to have younger patients, you might be able to go down to grade school ages. And have them self-collect, then we would want to see in usability studies that down to the age group that you're going to say, down to the age that you're going to say they can do it, that we'd like to see usability down to that age and good representation in the kids. If you're going to have adults collect, say kids under 14 or under 13, I forget what the exact cut point is, and you want to include, unlimited pediatric patients in your test, then we would want to see usability of adult collection down to age 2. And so obviously if you want to get a full pediatric range, you can involve some self-collection of older kids if you want, or it can be all adult collection, but then whatever, say it's, whatever grade 2, you demonstrate that kids can self-collect down to grade 2, then below grade 2, you're going to have adult collection, and we'd want to see representation that an adult in a home test situation or, that kind of setting can safely and adequately collect under usability studies, and also in your clinical studies, that adults can safely and adequately collect those samples, and those samples are apparently in the usability studies collected well, and in the clinical studies accurately. So that's a lot of information. It all depends on which route you want to go as far as what claims you want about self-collection for kids or adult collection. But that's what we've done for all the authorizations to date. And if there aren't kids included in the study, we can't authorize kids. And of course, in point of care situations we know that clinicians can adequately sample kids. So we're not worried about testing it necessarily in kids, but you shouldn't exclude kids in your clinical study at point of care. It's just we don't need to specifically see kids in the point of care study unless there's something unique about your device that makes kids difficult. Hopefully that's helpful.
Well, okay. Actually, what I'm looking for is that can we get if the device is approved for point of care, and now we work with you on the usability studies, depending on the age group, because it's massive. We're going after a massive population at large, and we can't just. I think this is beyond whatever any company's ability, small companies specifically, to do that kind of studies unless they work with you on the usability and allow the point of care device that's approved to be used to collect data. Is that possible?
No.
So
no, we're looking at usability in clinical studies that demonstrate performance in the home type situation, and it just can't be avoided. We make our decisions based on data, and if you only have point of care studies, you have healthcare sampled patients, healthcare workers sampled patients, and run by the healthcare workers. They are not sampled and run by home users.
Agreed. I'm asking if we have a point of care device, approved point of care device, and working with you on a usability study, design, and I will collect the data, and depending on the age group, is that going to be usable? The device can be used for that. So, and post-approval of point of care.
Yeah, I don't think we're actually communicating well in this forum. So I would suggest that you reach out either through a pre-EUA or the template email address because we're just not connecting on this. So I'm not understanding your question well enough to give you a response that meets your needs. And I'd like that to happen, but I think we ought to. If there's another caller, I think we ought to move on to the next caller.
Okay.
Thank you. Our next question will come from Elliot Rosen. Your line is open, sir.
Hello, good afternoon. I have a quick question regarding the serology test for neutralizing antibodies. I know on the town hall call last week, it was stated that due to declining prevalence in the United States, FDA would now consider data from subjects enrolled internationally. And my question is, will there be updated guidelines released for serology testing for neutralizing antibodies, or can we utilize the minutes from the call last week in case a reviewer asks about that during our EUA submission?
Yeah, you can certainly refer to the minutes, and they'll always, the reviewers, if they have an issue with what I said on this call or Toby says on the call, they'll come to me and try to understand it. So I don't believe I could be wrong, but I don't believe the neutralizing template says it has to be done in the U.S. We've just been discouraging non-U.S. Because prior to now, there's been unfortunately plenty of positive patients in the U.S. And given that our EUA bar is so much lower than the usual FDA test standard for numbers of positives and negatives and the amount of work done for an authorization, that we want to ensure that we best understand performance of that test on the relatively few samples we ask for in the U.S. population, in the U.S. labs and point-of-care sites and home sites. Due to the incredible success of the U.S. to date and right now, can't predict the future, the vaccination program and the testing program and all the other programs that everybody has been involved in, apparently has been highly successful, except for pockets right now, that we're in a bit of a conundrum, on test validation. And we're trying to be as flexible as we can. Toby, do you have anything to add?
Yeah, I just want to mention, I believe the template does specifically say U.S. And
Okay.
we want to make sure that that the specimens that are used reflect the population, that the intended use population. So we would just want to see that justification that we've talked about on this call as well, about when you're using, we're considering using OUS specimens, outside the US specimens, that we would want to see the justification for how that is representative of a US population, including the circulating variants that are in the US.
All right. Thanks, Toby, for that clarification and correction. And we're constantly looking at updates to templates that will be helpful. It does, and Toby's actually primarily the one responsible for putting all that together and marshaling it through all the authorizations within the U.S. government, and she does a great job. But it can take some time, and sometimes things are in process, and we don't want to slow what's already in process to update. So that's the incredible value, I think, of this constant weekly call that we have with developers, where we can update our most current thinking well ahead of what the templates reflect, and it's one of the main reasons why, one of the key reasons why we have this call, and why it's, I think, important for people to call in and or check the transcripts. Thank you.
Great, thank you so much. Is there a plan right now to update the template, or is that still?
I think it's a good thing to do, and like I said, we tend to accumulate updates and send them up at the appropriate times, because it is a lot of work to get all the authorizations before we can post it.
Understood. Thank you very much.
Thank you. Our next question is from Ariana Erickson. Your line is open, ma'am.
Good morning. I have a question about an antigen lateral flow test. So we're working on a point-of-care claim for our antigen test using a CLIA-waived drive-through clinic. At the clinic, they use personnel who are certified to conduct nasal swabs and nasopharyngeal swabs, and the same personnel will be collecting and running our tests. I just wanted to ask if you anticipate any issues with these personnel as part of our point-of-care claim.
No, but do double check the template we do want to see more than one individual, and we recommend more than one site be used point of care. But sometimes, it may be necessary to use one site. We want to see, I forget the number, I think it might be 5 or 6 different individuals, but at that one site, so that we have a spectrum of users, and so we're not just going with with somebody who's like expert, and we're not seeing the typical point of care site user.
Absolutely. Yes. Thank you so much.
Thank you. Our next question is from Robert DiTullio. Your line is open, sir.
Yeah, thank you. Hi, Tim and Toby. Thank you for taking my question. I wanted to follow up on an answer that I heard to one of the previous submitted questions about POC ex-US and qualifying the sites and the users. I heard the response that you should file an EUA to tell about the new study or the amended study. If the sponsor is already operating under a previously submitted EUA, and because of the dearth of samples now needs to pivot outside the United States, is the pre-EUA the right way to go? Because you're so busy, you don't get immediate responses. Is there a quicker way to verify something, like maybe via email or something like that, or do we need to refile a pre-EUA?
So if you've already filed a pre-EUA for a device and you're switching to this because you've tried a prospective study in the U.S. and it isn't getting you where you need, I think it's very appropriate to go back to the same reviewer who reviewed your pre-EUA and ask them this question. And you can just go directly to them. They can just reopen the file or reopen a new file and address that question specifically. For those that we haven't seen the technology yet at all, it's probably best to go directly through a pre-EUA process so that we understand the technology and we can address the questions, accurately. I know we still have a backlog of EUAs and pre-EUAs. I've made it a concerted effort to mention that we need to make good progress on both pre-EUAs and EUAs. The pre-EUAs are not required, and our recommendations in the EUA templates are not required. It's recommended, pre-EUAs are recommended, and our templates are recommended validations. The value of the pre-EUA, as just like the value of the Q-sub or pre-submission, it reduces the risk to the developer, and the reviewers are still just swamped. So we're doing our very best. Developers can do things on risk, and so it's why we give high-level guidance here on our recommendations. If it's point of care, we want to see it, exactly mimic the point of care situation in the U.S. So that's a busy clinical office practice or a busy ER. It's not a central hospital lab where you pull trained laboratorians off that line and say, oh, test some samples on this point of care device. No, that's not testing on, folks who aren't trained laboratorians. And we have seen those without a pre-EUA. We've seen sponsors say, oh, here's our point of care study. And, oh, by the way, we did it in a central lab with central lab trained laboratorians. And we're like, that's not point of care. You get a moderate high complexity claim, but not going to get a point of care claim. So that's the challenge. So if you can do a good job on selecting point of care sites or home sites, as we've talked about those situations already, this call and many calls, that can reduce your risk if you follow our high level guidance. But to eliminate, complete risk of having to repeat some studies, the pre-EUA process is still the best way that we recommend.
Okay, well, as usual, very helpful. Thanks so much, Tim. I appreciate it. And I wasn't impugning the process that it was too slow. I was just saying the reality is it takes a couple of weeks or so, and sometimes, and for developers, sometimes that's, a long time.
I know. I feel for you because I've spent many years, 15 plus years on the other side of the fence and development community and working with the FDA. And I know how important it is. And I track at least weekly all of the volumes for the various submissions and work with our team to do the very best job that we can to get answers as quickly as possible.
Yeah, thanks. Well, keep up the good work. Thanks so much. Bye-bye.
I will. Thanks.
Once again, if you would like to ask a question, please press star 1 on your touch-tone phone, and please record your name. We do have a question from Kamudi Venkat. Your line is open, sir.
Good afternoon. Thanks for taking the question. Thank you very much for all the hard work that your center is doing, and you are doing your best. But if the best is not good enough, and especially when you really don't have enough resources to do it, and is there anything that can be done better to increase the efficiency, make this process more transparent, to be more helpful for the developers, especially not big corporations who are small businesses, so that innovative methods and tests that can be made available during this pandemic?
We're doing our very best. We've authorized almost 400 tests now, and you don't hear about the ones that we don't authorize. And so we have plowed through a lot more tests than that. And we believe we're doing a great job at meeting the public health need at this point. And with all those tests authorized, and we have our priorities. Which is a point of care, particularly diagnostic tests, point of care, and in the home diagnostic tests, and home collections, and high throughput central lab tests. So those priorities haven't changed, for a very long time. We always assess priorities, and the team is doing the very best they can. We also moved 100 people from other areas recently onto COVID work. That surge and had great success in clearing the backlog in many cases for priority reviews, and but unfortunately they put more than 100 regular non-COVID 510K de novo and PMA submissions on hold, or on pause, not hold, on pause, and that got to be a balance of our resources. So our office was not staffed. The center was not staffed prior to this pandemic to handle this volume. Our office, again, I'll say it again, in the past year, we typically see about 1,900 applications a year. Our office alone received over 5,500 applications in 12 months, with no substantial increase in staffing because it takes experts to review this. So we just had to do it faster and better than we had ever done it before, which I believe the team did. And okay, I think it's time to move on to the next caller. Thank you.
We have no more questions at this